Welcome to the Deep Dive, where we plunge into the stories behind the knowledge that shapes our world.
Today, we're tackling a real medical game changer.
We really are. It's a tale of life-saving innovation, but it's not just about brilliant minds in labs. It's also about chance, some surprisingly untidy habits, and even, believe it or not, a bit of water polo.
Exactly. We're diving deep into Alexander Fleming and penicillin. You probably know the basics, the moldy dish story.
Right.
But we want to go further. Our mission here is to really unpack that journey, from that forgotten petri dish to a global phenomenon.
And look at the people often missed in the story, the ones who were absolutely key to getting penicillin out there.
And also grapple with its legacy. It was
Sure.
profound, obviously, but also challenging for modern medicine in ways we'll get into.
To do this, we've pulled from, well, all sorts of places. Biographies, historical docs, the original scientific papers, even some surprising cultural bits.
Yeah, trying to get that full picture for you.
So let's start at the beginning. Alexander Fleming, born 1881, Ayrshire, Scotland. A remote farm. How did that sort of rural start shape him, that curiosity?
Oh, it's fundamental, I think. Farm life, it demands observation. His parents, Hugh and Grace, they instilled that hard work ethic. And he was always curious about the natural world around him. His father died when he was only 7, and there were financial struggles. But his mother, Grace, was determined the kids got a good education. Those early observational skills were, well, crucial later on.
Okay, so he does well in school, but then hits a wall financially. Has to work as a shipping clerk in London at 16.
Yeah, not exactly the fast track to medicine.
So how does he pivot back?
Pure chance, almost. An inheritance from an uncle, John Fleming. That gave him the means.
Ah.
And then, here's the water polo connection. He was playing for the London Scottish Regiment, actually competed against St. Mary's Hospital's team.
No way.
Yes. And that connection, that's literally why he chose St. Mary's for medical school in 1903. It gets better. The captain of the rifle club at St. Mary's wanted to keep Fleming on the team.
Okay.
So he suggested Fleming join the research department to stay affiliated.
You're kidding. Rifle club recruitment leads to bacteriology.
Pretty much. That suggestion led him straight to Sir Almroth Wright, a pioneer in bacteriology. And Fleming, well, he excelled. Qualified with distinction in 1906, top medical student gold medal in 1908.
That is just an incredible sequence of events. Water polo and rifle shooting shaping medical history. So under Wright, he moves from wanting to be a surgeon to this new field, bacteriology. What was Wright's influence?
Wright was really thinking differently. He was big on vaccine therapy, boosting the body's own defenses, not just hitting microbes with external drugs.
Right, stimulating the immune system.
Exactly. That focus on internal defenses plus Wright's rigorous scientific method, it definitely honed Fleming's approach, that keen observation skill.
And we see that pay off pretty quickly, don't we? In 1921, he's got a cold.
Yes. The lysozyme story. He's nursing a cold. A drop of mucus literally drips onto a bacterial culture plate. Weeks later, he looks at it, and where the drop landed, the bacteria, gone, dissolved.
Wow.
That's lysozyme, a natural antiseptic in tears, saliva, mucus. A big deal for understanding immunity. Again, it shows how we notice things.
But lysozyme wasn't the answer for the really nasty bacteria.
No, it wasn't powerful enough for the really dangerous stuff.
Yeah.
But it was, another step. It sharpened his focus on finding something antibacterial.
And that focus gets tragically intensified during World War I. He's in France, makeshift lab, studying wound infections. What did he see there?
It was grim. He discovered that the standard antiseptics they were using, things like carbolic acid, they were often doing more harm than good.
How so?
They killed the body's own immune cells, its natural defenses, even faster than they killed the invading bacteria.
Oh, wow. So the treatment itself was dangerous.
In many cases, yes. Soldiers were dying from the antiseptics, not just the infections. Fleming argued for just keeping wounds clean and dry, letting the body try to heal.
Was he listened to?
Largely ignored, unfortunately.
Well.
The established practices were hard to change. But seeing that firsthand, the failure of existing treatments, it absolutely fueled his search for something better. He knew there had to be another way.
Which brings us finally to September 1928. Fleming's back from vacation. His famously messy lab.
Infamously messy, perhaps. He comes back, looks at a petri dish with staphylococcus bacteria that he'd left out, and sees mold, contamination. Specifically, a blue-green mold later identified as Penicillium notatum.
Okay, contamination happens, but what did he see?
He saw the pattern. Right around the mold, the staph bacteria colonies were dead, destroyed, a clear halo. Further away, the colonies were fine. His reaction.
That's funny.
Classic understatement. What made him notice that, do you think, when maybe someone else would have just tossed the plate? That inherent curiosity again, combined with his training. He wasn't just seeing mold, he was seeing an interaction, an effect. That zone of inhibition was the clue. So
Right.
he dives in, calls it mold juice at first. Then on March 7th, 1929, he formally names it penicillin, after the penicillium mold.
And he knew, didn't he? He said something later about that moment.
He did. He reflected, when I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine, but I suppose that was exactly what I did.
Amazing. And he also had that quote, one sometimes finds what one is not looking for.
Perfectly sums it up. Serendipity plus a prepared mind.
But okay, he finds it, names it, publishes in 1929. Why didn't it take off immediately? Why wasn't he the one to develop it into a drug right then?
That's the critical next step, or lack thereof, for Fleming. He was a brilliant bacteriologist, but not a chemist. He struggled to isolate and purify penicillin. He found it very unstable.
And he made a mistake, didn't he, about how it dissolved?
Yes. Crucially, he reported it was insoluble in certain solvents like ether, which turned out to be wrong. That made purification seem even harder. He basically concluded it was too fragile, acted too slowly in the body, and probably wouldn't be useful for treating infections.
So he kind of gave up on the therapeutic potential.
Largely, yes, on the chemical development side. He published his findings, which was vital, but didn't pursue the drug development pathway himself. He did, however, keep the mold culture alive and sent samples to anyone who asked. That turned out to be incredibly important.
Planting seeds for the future. So penicillin sits there, kind of dormant knowledge for almost a decade. What brings it back into the spotlight?
Well, interest in these kinds of natural antibacterial agents was low for a while. But then 1939, Oxford University. A biochemist named Ernst Chain reads Fleming's forgotten 1929 paper.
And he sees something Fleming missed, or sees a way forward.
He saw potential. He convinced his supervisor, Howard Florey, an Australian pathologist, that reinvestigating these microbial substances could be really promising. Florey agreed and assembled this amazing team.
The Oxford team. This wasn't just one person anymore.
Absolutely not. It was truly interdisciplinary. Florey leading, Chain handling the biochemistry, Norman Heatley, a fungal expert, crucial for figuring out how to grow the mold and measure its potency. They got a sample of Fleming's original mold strain, which had luckily been preserved at Oxford. This teamwork was key.
So they had the mold. Now they needed to make enough of it to actually test and purify it. In wartime Britain, no less. How did they manage?
With incredible ingenuity and resourcefulness, they grew the mold on liquid spake ox medium. At first, in anything they could find, flasks, bottles. Later, they used specially made ceramic vessels, almost like bedpans, just to get enough surface area for the mold to grow.
Bedpans for penicillin production?
Whatever worked. Purification was the really tough part. Using solvents like ether, later amyl acetate, Heatley developed a clever reverse extraction method using sodium hydroxide. It was painstaking work.
And Heatley also figured out how to measure how strong the stuff was, right? Since it was so unstable.
Yes, that was brilliant. He developed the penicillin assay, put samples on an agar plate with bacteria, and the bigger the clear ring around the sample where bacteria couldn't grow, the stronger the penicillin. That gave them the Oxford unit, a standard measure.
So they could finally quantify it. How potent did it seem?
Amazingly potent, even in impure forms, effective at like 1 part per million. Florey calculated it was maybe 20 times stronger than the best sulfa drugs they had then. That gave him the green light.
To move to animal trials must have been tense.
Hugely tense. May 25th, 1940, the famous mouse experiment. Florey injects 8 mice with deadly streptococcus. 4 get penicillin, 4 don't.
The result.
By the next morning, all the untreated mice were dead. The 4 treated with penicillin, all alive. Chain apparently just called it a miracle.
Wow, I can imagine. So human trials next. Very cautiously, I assume.
Very. First, just a safety test in January 41 on a woman, Elva Akers, who had terminal cancer, just to see if it was toxic. It wasn't. Then the first therapeutic attempt. Albert Alexander, the Oxford policeman. He had a terrible infection, sepsis spreading from a scratch.
Yeah.
He got the penicillin.
And it worked.
Dramatically. He started recovering. But they didn't have enough. They were even recycling penicillin from his urine, trying to purify it again. But it wasn't enough. The supply ran out, the infection came back, and he died.
Heartbreaking, but a crucial lesson learned.
Absolutely. It proved penicillin worked in humans, but it hammered home the desperate need for way more production. It also made them focus initially on treating children who needed smaller doses.
Were there similar things happening across the Atlantic in the U.S.?
Yes, concurrently.
Mm,
Martin Henry Dawson's team at Columbia were treating patients by October 1940, Aaron Alston and Charles Aronson.
right.
And then a really significant case, Anne Sheath Miller, March 1942. She had overwhelming streptococcal sepsis, was near death. Penicillin saved her life. She became the first American civilian successfully treated.
These results must have started making waves.
They did. Ethel and Howard Florey published the clinical trial data in The Lancet in March 1943. That really put penicillin on the map scientifically.
Which leads us right into the war context. World War II raging, infections killing soldiers left and right. The timing seems almost fated.
It really does. The need was immense. Gangrene, pneumonia, infected wounds. Penicillin looked like the answer they desperately needed. The war provided this huge, urgent impetus.
So Florey and Heatley head to the U.S. in 41, seeking help with mass production.
Yes, because Britain just didn't have the industrial capacity, especially under wartime conditions. They needed American industry. And that story about Heatley wanting to smear mold spores in their coat pockets.
Yeah, incredible, just in
It
case.
shows how precious they knew this stuff was. They couldn't risk losing it.
So America steps up. What were the breakthroughs there that cracked the mass production problem?
A few huge things. First, finding a better mold. Charles Thom at the USDA. He not only correctly ID'd Fleming's mold, but his team found a different strain, Penicillium chrysogenum. Where? On a moldy cantaloupe from a market in Peoria, Illinois.
The cantaloupe again.
Yep, and this Peoria strain, it produced 6 times more penicillin than Fleming's original.
Wow.
Then Andrew Jackson Moyer, also at the Peoria lab, found that adding corn steep liquor, basically a waste product from making cornstarch to the growth medium, boosted the yield another tenfold.
Corn waste product. Amazing. Finding value where no one else looked.
Exactly, and then came the engineering leap, deep tank fermentation.
Instead of those surface cultures in bottles and bedpans.
Right. Companies like Pfizer had experience with fermentation for things like citric acid. They adapted it, figured out how to grow the mold in huge, aerated, agitated tanks. Suddenly, you could make enormous quantities.
This U.S.-UK collaboration sounds unprecedented.
It really was, driven by the war. By September 1943, they'd ramped up production enough to supply the Allied forces. Compare that to late 42, when they barely had enough for maybe 100 patients total. Just a massive scale-up.
While the Allies were scaling up, what about other places? You mentioned efforts in occupied Europe, trying to make it secretly.
Yes, these are fascinating side stories. In France, Institut Pasteur and Rhône-Poulenc made small amounts and cleverly gave the Germans a useless strain when asked.
Deception in the name of science. And the Netherlands story is even wilder.
Absolutely. The Dutch Culture Collection, CBS, they had Fleming's strain hidden away, also gave the Nazis a dud. Meanwhile, a company, NGNSF, secretly made their own version, codenamed Bacchanal, from a different penicillium strain they had.
And the story about keeping their German guard drunk?
Apparently so. To keep their secret production under wraps, it was risky, clandestine work. And because they developed it independently, they weren't caught up in the later patent disputes.
Did the Germans ever manage to produce it themselves?
They did eventually, by late 1944. Hitler himself supposedly received it after the assassination attempt that year. But Allied bombing raids hampered any large-scale German production.
So by D-Day 1944, penicillin is widely available for Allied troops. Its impact must have been immediate.
Transformative. Infections like gas gangrene, a huge killer in World War I, there's something like 15% mortality for those wounded. It practically vanished among Allied troops who got penicillin. It really earned that miracle drug label on the battlefield.
But this success and the way the story got told led to the Fleming myth. Explain that.
Well, after a high-profile success story in 1942, treating a patient with meningitis using the Oxford penicillin, the Times ran a big article. Fleming's old boss, Sir Almroth Wright, really played up Fleming's role.
And Fleming himself was okay with the attention.
He seemed quite comfortable talking to the press, yes. Florey, however, actively disliked publicity. He forbade his Oxford team from giving interviews. He wanted to focus purely on the work.
So Fleming became the face of penicillin, even though the Oxford team made it usable.
Exactly. The public narrative became heavily skewed towards Fleming as the sole genius discoverer, overlooking the absolutely vital development work by Florey, Chain, Heatley, and the entire Oxford team, not to mention the American production effort.
Did the Nobel Prize set the record straight?
To some extent within the scientific world, yes. The 1945 Nobel Prize in Physiology or Medicine was awarded jointly and equally to Fleming, Florey, and Chain. It explicitly recognized them all for the discovery of penicillin and its curative effect.
And later, Dorothy Hodgkin won for figuring out its structure.
Yes, in 1964, Nobel Prize in Chemistry for using X-ray crystallography to determine the exact chemical structure, another crucial piece of the puzzle.
There was also controversy about patents, wasn't there? Florey versus Chain.
Right. Chain felt they should patent it, secure the discovery. Florey was adamant it should be a gift to humanity, no patents. A real ethical clash.
But patents were filed in the U.S.
Yes. Andrew Jackson Moyer, who developed the corn steep liquor method, and the company Merck filed patents on the production processes, which led to this slightly absurd situation where British researchers later found themselves potentially paying royalties to U.S. firms for aspects of a British discovery.
That must have stung.
It definitely highlighted the need for better management of national scientific discoveries, leading to the UK creating the National Research Development Corporation, the NRDC, in 1948.
Despite all that, Fleming received immense recognition beyond the Nobel.
Oh, absolutely. Knighted in 44, head of the Wright Fleming Institute, president of the Society for General Microbiology, member of the Pontifical Academy of Science. Honorary everything, basically.
And some unusual honors, too.
Definitely. Honorary chief of the Kiowa tribe in America. Nearly 30 honorary doctorates. Voted third greatest Scot on Time magazine's 100 most important people of the 20th century list.
And you can still visit his lab.
You can. The Alexander Fleming Laboratory Museum at St. Mary's is preserved. Plus, there's the Fleming Museum in Greece, set up by his second wife, Amalia, preserving notebooks, photos, artifacts, and those statues. Madrid matadors were apparently very grateful.
Reduced deaths in the bull ring, right?
And squares, schools named after him, his name is everywhere.
So penicillin changes everything, saves millions. But Fleming himself saw a potential dark side, didn't he? Even back then.
He absolutely did. It's quite remarkable. In his 1945 Nobel lecture, he gave this stark warning. He said overuse, and especially underdosing, could lead to bacteria becoming resistant.
He predicted antibiotic resistance right at the start.
He specifically warned that if you expose microbes to non-lethal quantities, you essentially teach them to resist the drug. Prophetic.
And how quickly did that prophecy start coming true?
Disturbingly quickly. Even before mass production, in 1940, Chain and Edward Abraham found an E. coli strain that produced an enzyme, penicillinase, that broke down penicillin. First sign of trouble.
Before it was even widely used.
Yes. By 1942, resistant Staphylococcus aureus strains were showing up. By the 1960s, many common staph infections were resistant.
What drove this rapid resistance? Just overuse in medicine?
That was a huge factor. Penicillin was seen as a cure-all. People wanted it for colds, flu, things it doesn't even treat because they're viral. Doctors felt pressured to prescribe. It was used prophylactically in hospitals, sometimes unnecessarily.
And then there was the agricultural use. That seems like a big, maybe unexpected contributor.
Hugely significant.
Yeah.
Post-war, they found adding small amounts of penicillin to animal feed made livestock grow faster and controlled some diseases. The FDA approved this in 1951.
So constant low-level exposure for bacteria in farm animals.
Exactly. Breeding grounds for resistance. Plus, you had issues like penicillin residues showing up in milk, messing with cheese production, causing allergic reactions in people.
Did regulators eventually catch on to the danger of agricultural use?
It took time and, sadly, often tragedies. In the UK, the Netherthorpe Committee initially relaxed rules, but then a terrible incident in 1967. 15 children died from a multi-resistant E. coli outbreak linked to farm animals that really forced the issue.
Right.
The UK banned non-therapeutic antibiotic use in animal feed in 1971. Other European countries followed. But the genie was kind of out of the bottle.
Leading to the era of superbugs.
Yes. By the 70s, and definitely accelerating into the 2000s, we saw the rise of bacteria resistant to multiple antibiotics. MRSA-resistant tuberculosis, drug-resistant gonorrhea. Infections that were once easily treatable became dangerous, sometimes untreatable again. The WHO now calls antibiotic resistance one of the biggest global health threats we face.
It's a scary prospect. And you mentioned the dry pipeline. It's hard to develop new antibiotics now.
It is. Economically, it's tough for pharmaceutical companies. Developing a new antibiotic is expensive, takes years. And if resistance develops quickly, which it often does, the drug doesn't have a long, profitable lifespan compared to, say, a drug for a chronic condition.
So the financial incentives aren't really there?
Not as much as needed. So the focus has shifted. It's partly about preserving the antibiotics we do have.
How do we do that?
Stricter prescribing guidelines, antibiotic stewardship programs,
Yeah.
educating doctors and the public about proper use, finishing courses, cracking down further on agricultural misuse, basically using them less and more wisely.
And what about finding new ways to fight bacteria beyond traditional antibiotics?
That's the other major focus. There's renewed interest in things like phage therapy, using viruses that naturally prey on specific bacteria.
Viruses to fight bacteria.
Exactly. Also, immunotherapy, finding ways to boost our own immune systems to fight infection better.
Mm.
And using cutting-edge tech like AI and genetic engineering to maybe design entirely new classes of drugs or tailor treatments. It's an ongoing arms race, really.
So we've traced this incredible arc from that funny mold on a plate
Yeah.
to a drug that saved millions, changed warfare, changed medicine fundamentally, but also a story that carries this heavy warning about resistance.
It's such a powerful lesson, isn't it? It shows the brilliance of discovery, the absolute necessity of collaboration. Fleming found it, Oxford developed it, industry produced it, but also the critical need for responsibility, stewardship.
Because Penicillin's story isn't over, it's still unfolding. With this challenge of resistance, it really makes you think even our biggest medical wins aren't permanent victories.
They require constant vigilance.
So, the provocative thought for you, the listener, reflecting on this whole journey, the chance discovery, the wartime urgency, the miracle cures, and now the resistance crisis, what does Penicillin's story teach us about our relationship with the microbial world? And how do we ensure that the power of antibiotics, that miracle, isn't lost for future generations?